This is the preparation for lecture 16. We're going to cover two basic topics. One is the maintenance of the shoot apical meristem, and the other is a little bit about the patterning and more about the exact placement of leaves as the meristem is giving rise to new shoots. So I wanted to show this because when we see these schematic diagrams, we're actually, they're representing this. So this is an actual real meristem, and it's a slice taken through a developing plant. So it's a mature plant, and it's growing. And the leaf primordia are shown on the sides here. This is what they actually look like, and you'll see them in the schematics that we show. And then this little area here is the area that's shown right here. Okay, so there's the central zone. This is the, oops, that's not a good start. So the central zone is this part that's shown in red. And there is a region below here that's sort of the area where stem cells are induced and maintained, and there's some signaling that goes back and forth in this area. The peripheral zone, this part here, the blue parts, that is where the new leaf primordia are going to develop. So as this continues to grow, there's a fairly mature leaf here, and so this is going to give rise to the next leaf primordium, which will kind of bulge out from here and then start to grow. And then as it shoots up even further, they'll be in another position, another leaf primordium. And then down in this area is what's called the rib zone, and this is where the vascular tissues and so on are going to form. So this is just to get you oriented, just so you can sort of understand what you'll be looking at. This is a real shoot apical meristem, and it has been false colored by a computer, so this isn't real staining. And the dark is the beginnings of a new leaf primordium. But the thing I want you to pay attention to here is just that there are these layers. So L1 and L2 are each one cell wide. So it's just a single layer of cells on the surface, and L1 is going to form the epidermis. So that's the outer skin, if you like, the outer coating of the plant. And then L2 are the cells immediately below that. And those are going to mostly produce leaves or flowers if it turns into an inflorescence meristem, which we will be talking about in the next class. And then the cells under here are mostly giving rise to the stem. And one thing to pay attention to is that when you look at this schematic, is a little bit easier to see, although you can kind of still see it in this image. If you look, the L1 and the L2 layers, they're very well organized. They're one cell thick, and they're the plane of division. So the cells are all oriented sort of right next to each other. And when divisions occur, they occur in this anticlinal way. So they are perpendicular to the surface. So if that's the surface of the cell, then anticlinal is division in this direction, right? If you look at the cells underneath, notice a couple of things. They're very different in size, and the cell walls, they're not sort of neatly kind of almost rectangular or almost square in some cases cells. They have a variety of different shapes, different sizes, and you can see that planes of division have occurred in many different directions. And so the organization of the cells is not as strict as it is on the outer layers. These two layers, the neat and tidy anticlinal ones, those are called the tunica. And then below that is the corpus. Corpus just means body, and it's basically everything except the outer coat, which is the epidermis, and then the cells that are going to give rise to the leaves on the stem. Okay, and so in talking about shoot meristem development, SAM, we talked about the shoot meristemless stem gene last time. The mutant phenotype is having no shoot apical meristem. And the function of this gene product, the protein, is that it prevents the stem cells from all differentiating. And so usually for any kind of stem cell, you will have stem cells that they will divide. And we talked about this a little bit when we talked about the oogenesis in flies, so that's a connection back. Plants have the same kind of features. They will have one daughter cell that goes on to differentiate. It forms a structure. It contributes to leaves. It contributes to flowers or something. But the other daughter cell will be a stem cell as well, so that you don't run out of stem cells. And the problem with stem mutants is that they do actually run out of stem cells, because there's nothing that stops this cell from also differentiating. And now both of the daughter cells of this stem cell, they form, the beginnings of leaves and so on, and then the plant just runs out of stem cells. So if you go back and take a look at the mutant phenotype of that loss of function mutation, you will see that. The phenotype matches the function. So this is about maintaining stem cell identity. Now the other two genes, or three genes, I guess, that we're going to be talking about, or four genes, are Wuschel. And Wuschel is, its main function is to promote the formation of stem cells. So when you've got mutants in Wuschel, you have the loss of stem cells, okay? So it has sort of a funny bushy phenotype, but the underlying reason for it is the loss of stem cells. And so it's promoting stem cells. The clavata genes, there's a ligand and then a heterodimer receptor, and we're going to talk about those as well. And if you have a mutation, then the shoot apical meristem gets larger. Because there's additional stem cells. So you've got Wuschel promoting stem cells, and then clavata pushing back and reducing the stem cell population. And through the interactions of these two sets of genes with their antagonistic roles, they keep a consistent and reasonable size of stem cells, right? So if you have a mutant in one or the other, the mutant phenotype tells you what that system is supposed to be doing, and that's kind of the key here. So Wuschel is a specific homeobox containing transcription factor, so it's similar but has somewhat different role than sort of homeoboxes that we talked about when we talked about the bithorax complex, for example, in flies. So it's a transcription factor. One of the genes that it promotes is the CLAV3. In response to Wuschel, CLAV3 gets expressed. And CLAV1 and 2, clavata, these are two proteins that are heterodimers. So they make a cell surface receptor. And the protein, they're two different proteins, and they dimerize to form the overall receptor, and then CLAV3 is the ligand for that. Okay, and so this is a picture from Current Biology. Current Biology is a terrific journal, and it has wonderful sort of reviews and overall articles that explain particular topics. So just for orientation, this is the central zone here, and that's where the stem cells actually are. But underneath, this is a kind of a slight overlap region, and this is the organizing center. And so it's kind of in this area that Wuschel is expressed. And then just to note, this is the peripheral zone. So it's this is analogous to the other image that I showed, but I like the way it's kind of shown. And this is where the new leaf primordia are going to develop. But then as the shoot apical meristem continues to grow, because of course the plant is growing at the time, and then as the new leaves are formed, they remain on the stem, and the shoot apical meristem is, it's advanced. So just to sort of get oriented. So these are they're called primordia because they haven't fully formed a leaf yet. They're still kind of a bulge, and we'll talk about the flattening of them and the patterning of them later on. And then up in this region, so Wuschel is transmitted. It travels up here, and it causes CLAV3 to be expressed up in this region here. And the cells surrounding here have the receptors CLAV1 and 2 on both sides. I'll just draw it in one. And so CLAV3 is a diffusible ligand. It's able to move and bind to the receptor here, and the receptor has the downstream effect of actually repressing Wuschel. Now this isn't beautiful here because I have whited out a couple of genes that I think we'll leave out. I would just, if you remember just this part, I think that's sufficient for our purposes. So hopefully that makes sense. And so how do we know what these different genes are doing, especially before we had them all fully sequenced and had isolated the proteins and so on? It turned out with Wuschel, if you stain for the presence of Wuschel, it's found in these. So this is that region a little bit below the central zone. So that's the central zone, and this is the peripheral zone to either side. And up here is where the stem cells are. But if you make a CLAV3 mutant, then you find that Wuschel expression expands across to up into these regions here. And you can't, it's not easy to see because this is a three-dimensional structure and a two-dimensional picture, but there's Wuschel, everywhere. It didn't move to new positions and leave the old position. It's still down here, but it's also, you can see it right in those two outer layers, right? So when CLAV3 is mutated, then Wuschel can expand. So therefore, we can understand then that CLAV3 restricts or limits where Wuschel can be expressed. Remember that Wuschel is promoting stem cell fate. So the purpose of CLAV3 is to make sure that you don't have too many cells that are specified to be stem cells. You need to keep them within a reasonable limit to make the development work correctly. And so here are some others. So the Wuschel expression is expanded in the various CLAV mutants, CLAV3, 1, and 2. And if you make mutants in Wuschel, then you find that CLAV3 expression actually drops. And so that means that while CLAV3 and the other CLAVs, clavatas, are repressing Wuschel expression, Wuschel is upregulating CLAV3, right? So Wuschel is expressed in the cells below the central zone. And when you have mutants in Wuschel, you are missing that central zone. So the stem cell identity doesn't form in that region. Okay, so when you put all of these kinds of observations together, then this is what you get. So in the organizing center below, you have Wuschel expression. It basically leads to the expression of CLAV3. Okay, and then CLAV3 is a diffusible signal, and these cells have been induced to have a stem cell fate. Okay, and part of being a stem cell is to express CLAV3. And this can move laterally and downward. Okay. And the cells surrounding the Wuschel, so these guys, all these cells are protected, but the cells nearby have the receptor. Never mind about this one, we'll just talk about CLAV1 and CLAV2. And when CLAV3 binds to this receptor, that causes Wuschel to be repressed in these regions. And that means that Wuschel can only be expressed in its little location in the central area, and therefore it's kept restricted. So you've got Wuschel busily promoting stem cell production, but the stem cells themselves produce a repressor, and when these two things are in balance, you have just the right amount of stem cells in this region. You've got a, yeah, balance is the right word. So hopefully that is clear. I'm just gonna take some of this messy stuff away so you can read the slide better. Okay, so that's the little story of how that shoot apical meristem is maintained. The size doesn't change. As the shoot is growing, the meristem is about the same size right at the tip, the growing tip, and the leaves are forming, and the shoot apical meristem remains a constant size as the growth is occurring. And so this is a schematic. This is just the shoot apical meristem region. You don't want an eraser there. So this would be kind of the central zone where all the action is happening. When a new leaf has been stimulated to develop, it starts as a little bit of a bulge. And then over time, there's growth. So this is, further up the stem because the stem has grown. It's kind of hard to show that with an image. And the first leaf primordia is growing out more. And then the second one forms, and it forms in a precise location relative to the first one. And then even further growth, the first leaf primordium now is flat on one surface and rounded on the other. And we'll talk about that. It's ab and adaxial polarity. The structure on the surface and the bottom of the leaves are somewhat different. The second one is growing out, and then here's the little bulge where the third one is going to form, again, a certain distance from the second one. And the continued growth is happening. So there's a bunch of different things happening at once. There's always the new primordium starting to develop as the older ones are growing out in a proximo-distal direction, which I'll talk about in a minute, and they're being patterned ab and adaxial. Okay. So this is just, think about proximal being your shoulder and distal being your hand. Distal means it's far away from sort of your body, right? Your torso. And the same with the plant. The proximal is at the stem, and the distal is sort of the tip of the leaf. And then the dorsal is adaxial is the term. And the ventral, the underside of the leaf, is abaxial. Now, these are not easy terms to remember, and you will not have probably heard them before. I always remember B for bottom. So the B is the bottom or the underside of the leaf. Sometimes these little tricks can help you remember terms, especially at the beginning when they're not very familiar. And so if we were to look at a cross-section of a leaf, the adaxial surface is usually, it has a sort of a cuticle, and it often has a waxy coat on the top. If you look inside, you can see that a lot of the cells in the palisade layer here, these are your photosynthesizing cells. They have lots and lots of chloroplasts, and they are up near the surface because, of course, when the sun is shining, the sun is going to come here, and so you want your photosynthesizing cells up close to there so that they can collect the sunlight and produce the sugars. On the undersurface, you have these stomata, which are little openings that can open and close to allow for gas exchange. You have to have gas exchange because you can't photosynthesize unless the CO2 comes in to be fixed, and the oxygen is released as a byproduct. And also the stomata can open and close to moderate water loss. So if it's a very hot and dry conditions, the stomata can close so the plant doesn't lose very much water. And then when conditions improve, then the stomata can open again to allow for gas exchange. So the point here is not for you to memorize all of this, except just to notice that functionally it makes sense for the stomata to be on the underside. You don't want the sun beating down on this surface and then have your water evaporating away. A waxy layer to protect from water loss and so on. And then the structures inside kind of reflect the different function, where there's photosynthesis here and gas exchange there. Horribly oversimplified, but that's the general idea of the different patterning. And as the leaf primordia are developing, they tend to be very flat on the adaxial side and more rounded on the abaxial side as they develop and before the real flattening of the leaf takes place. So if you were looking at the development, you can see like they are a bulge first, and then there's this sort of curve, but as they get bigger, then there's more of a difference between the adaxial, this is really the dorsal side, and the abaxial, and the abaxial is more curved. Okay, so we're going to talk about that patterning, but then we're also, if you look at this this image, you notice there's a very precise spacing of the leaves. And so how does the plant kind of know or decide where those leaves are going to be placed? Okay, so these are the genes to understand regarding the patterning of the leaves. And I'm just remembering, if you there's one of the earliest slides, there's some staining of very dark brown, and I've written FIL, filamentous flower, beside it because that's one of the earliest genes that is expressed, and it affects both the patterning of flowers but also the patterning of leaves, and it's required for abaxial fate. So it's a transcription factor. If you were going to look at a leaf primordium as it was developing, with the sort of flatter side on the adaxial, then you would find that the filamentous flower protein was found on the abaxial side. Okay, and if you overexpress it, then what you find is as the leaf develops, it's abaxial all the way around. So you've got stomata all the way around on both surfaces of the leaf. If you make a loss of function mutation of it, then there's no abaxial specification and you don't have stomata, or you have very, very few, and that's often lethal. So you can make an overexpression of this gene and still have the plant survive because you've got it in like greenhouses, right, where you're providing really good conditions. And then there are 3 transcription factors that are responsible for adaxial fate. They're called fabulosa, favoluta, and revoluta. So, and they're very closely related. They all do pretty much similar jobs. But what's interesting about them is that if you make a mutation in any one of these, you lose adaxial fate. So you have to have all 3 functioning, and they can't cover for each other. So if we make a revoluta mutation, then the leaves are abaxial all the way around. So again, you've got stomata all the way around, and you don't have that waxy coating that the adaxial surface normally has. So filamentous flower as I said, is expressed in this sort of abaxial region down here. Fabulosa, Favoluta, and Revoluta, they're all expressed in the adaxial position. And if you lose the function of this, then it's abaxial all the way around. If you overexpress this, it's abaxial all the way around. Okay, and that's really, for this part, all that you need to remember. And then finally, we're going to go back and look again at the positioning of leaves. And in doing this, we'll be taking a look at auxin function. And so there's different types of phyllotaxy. There's whorled phyllotaxies, where you have a certain angle difference between this, and then between that, and so on. And they can have single leaves, they can have 2 leaves per whorl, they can have 3, and you've probably seen plants that are like this. But there's also phyllotaxy. Phyllotaxy just is a fancy word that means sort of what the pattern of the leaf placement is. And the one we're going to look at is the spiral phyllotaxy. And that is where single leaves emerge, and then the next one is 137.5 degrees around the circle from the first one, and then the next one is 137.5 degrees from that one, and so on. So there is this consistent angle all the way around. And so the numbering, this is the first leaf that developed, and then the second, and then the third, fourth, fifth, sixth, and you can follow it along and just see that the emergence of the leaves is at this very precise, regular pattern. And I've already kind of mentioned this, so I won't spend too much time except just to remind you that the primordium emerges as sort of a bulge in the peripheral zone of the meristem, but then there is this proximal outgrowth, so it extends outward away from the main central part of the body, and then there is this flattening where you have it more curved as it's developing on the underside, and flatter on the upper side. So that's the basic process that we're looking at. And there's some interesting experiments. So if you have a leaf primordium, and you've got some of the subsequent ones, so this would only be a bulge, and this would be just a little bit of outgrowth. This one's starting to flatten a bit. But if you actually excise from the developing meristem a leaf primordium, an older leaf primordium, the tissue, if you're very careful in how you work, this tissue will close up. And when this happens, then there's a shift in where the next leaf forms. So instead of being over here, it shifts over. And this, when this kind of observation was made, they thought that it's possible that there is some kind of inhibition coming from this older leaf primordium that's preventing this, leaf number 4 from developing. And when you remove this, if you remove I'm pointing with my hand, which doesn't make any sense, but when you remove this, you're removing that inhibition, and so that allows for the development of the new primordium in a place that's not the normal place, further along. And so this gave rise to this idea of some kind of inhibition, right? So this was the model. How do you space out your leaves? So if each of the primordia, each of the little baby leaves that's developing is the source of some kind of inhibition, think of a diffusible signal, because this is what everyone thought at the time, and that because with diffusion the concentration gets less, so the inhibition lessens as you move further away. And so the next time you reach a place where there's not enough of that inhibitory signal, we call that the next available gap. And so when the cells are in a place where that inhibitor is not there anymore, those guys can go ahead and form a leaf. It turns out, though, that it is auxin that is involved with this, and it's not actually a diffusible inhibitor, but it's sort of the opposite of that. So the newly formed leaf primordia, somehow all the auxin is transported. They kind of hog it, basically. They're sequestering it. And so what that does is it leaves what we call a zone of depletion, a region around the new leaf that hasn't got any auxin, and you cannot develop a leaf if you don't have any auxin. So really what we're looking at is not a diffusible inhibitor, but a lack of auxin being the inhibitor. Okay, so we're going to go through this. This is the current model. And so here's your older leaf, and you've got high auxin here, and there's less here. And then here, where there's almost zero, the auxin is transported away from the low concentration. This is a more recently started leaf primordium, and the auxin has transported in this direction to form another little, what we would call local maximum. Okay, and so the leaf is forming there, and the auxin is being sequestered there. But further away, there isn't any auxin, so it gets transported again, sort of in another direction, and it all starts rushing in this direction. So we'll go through this properly, because I tried to sort of organize it. Okay, so auxin is moving towards that higher concentration, and that's basically what's happening. So you're looking at maxima, local regions where there's tons of auxin, and then the area right around that maxima is completely depleted, because all the auxin is always being moving towards its higher concentration. Okay, so I should just have these here. So this one has stopped depleting the auxin, but this one has started to deplete it. And then in regions where there's less, it moves away, right? And so this is the new maximum that formed. And then this leaf will start sequestering the auxin, and that will create a zone of depletion around it, and it will cause the auxin to then move in a different direction away from that depletion zone. Okay. So P3 is depleting the auxin now. There's not very much of it, and so what little bit there is in these areas is being transported away. So any place where you've got these depleted zones, areas where the auxin is low, then the transport moves the auxin away. And when that happens, then there will be different directions of transport, but basically there will be a place where a number of transport from a number of different regions converge and creates this new maximum. And that's kind of the idea. So hopefully this makes sense. Something like this is the kind of thing you might have to talk your way through a few times to get a good sense of it. Most of this is incredibly unfamiliar. And also just give some thought as to, if the auxin is going this way towards this maximum, and then once the leaf primordium has depleted and made this zone of depletion around itself, now the auxin gets transported away. How does that happen? So think about how does it suddenly shift and change its direction? Okay, and then one of my favorite topics is how flowers develop and how flowers are patterned, and that will be the subject of our final lecture on plants.